Welcome, everyone. We'll go ahead and get started. It's a special pleasure to introduce Mark Noble. I'm going to read you one tiny little bit of his CV. So Mark is a professor of genetics, biomedical genetics, and neurosciences at the University of Rochester. He's the director of the University of Rochester Stem Cell and Regenerative Medicine Institute, and he's the Martha Freeman Distinguished Professor of Biomedical Genetics, and that's all I'm going to read off of his CV. So reading off his CV, it's long and distinguished, and you'd be impressed, but that's actually kind of doesn't really capture Mark. So there's three things I need to tell you about Mark that, in my view, make him really special. The first is his originality. Mark is absolutely one of a kind, both as a scientist and as a human being. And for those of you that are fortunate enough to interact with him, you'll find this out for yourself. But when it comes to any aspect of science and any aspect of the world around him, Mark takes his unique view and applies it in the way that he feels is intellectually honest. I can guarantee you that when the herd is going this way, Mark is going that way. Not to be contrarian, but simply because he's such an independent thinker. That's one of the things I really admire about him. The second thing I need to tell you about is his breadth. So I have this kind of romanticized idea that there was a time when scientists, sometime decades ago, were these incredibly broad thinkers, and they were erudite and thoughtful about everything. And these days, I think we make the excuse that the world is so complicated that we all have to focus down on our little subspecialties and so forth. But I guarantee you, whether you're talking about neuroscience or philosophy or music or art or history or any other topic or literature you care to engage him in, Mark is equally adept at talking about any of these topics, and really interestingly, he's thought about each of those topics and has original ideas, and I find that remarkable. It's a true Renaissance person, and it inspires me to be more like that. I think it's quite extraordinary. And the last thing I would tell you about Mark is about his generosity. Intellectually and personally, he's one of the most generous people I've ever met. I had the great good fortune of being his colleague in Rochester for over a decade, and he taught me probably more about science than anybody I've ever met. And so it was with tremendous great pleasure that I welcome him here today.
Wow, thank you, Craig. I'm so glad that I decided not to talk about my interest in the influence of the French Baroque architecture period between April and May of 1894 on the later development of stem cell biology. That was so kind. I was also so worried that you were going to tell the stories about Lindsay Lohan, which are not true. Mostly not true. One of them's not true. I want to cover a lot of ground. This is not going to be either your usual seminar or my usual seminar, because I have not been wanting to give talks for some time because I didn't feel that I was ready to talk about what I'm really interested in. And now I hope that we have enough of this work out there so that I can talk about what I'm really interested in without having you wonder about either my sanity or Craig's sanity for inviting me. So what intrigues me are very large biological questions. How do cells work? How do cells do the right thing at the right time? But I'm also interested in medical questions about how are we going to do a better job at developing therapies. And I've been particularly interested in this due to several years of being involved in reviews at the California Institute of Regenerative Medicine, where the first $2 billion that were spent led to about 5% of the projects actually getting to an IND stage. Yet this is an effort that is completely devoted to a translational purpose. That 1 in 20 rate is, of course, far better than the NIH, which funds a lot of basic research. But the fact is, it is not that different from pharmaceutical companies. It's probably better than biotechnology companies. So is it the case that we are stuck, no matter what we do, at a 1 in 20 probability that anything that we do is actually even going to lead to a clinical investigation. The costs of therapeutic development are so high. People talk about $1 billion per drug. They're starting to talk about $2 billion per drug, which means that the tolerance of failures is very low. And considering that 9 out of 10 drugs that enter a phase 1 trial fail usually before the phase 3 trial is completed, that's an extraordinary barrier to development of therapies. So, can we improve on this low transition probability? Can we increase success probabilities while also decreasing costs? And how do we do this in a way that addresses the urgent medical needs of the patients? In thinking about this problem, one of the general areas that one needs to consider, particularly these days, is where you're going to place yourself on the spectrum of personalized medicine to general principles. Now, there's a lot of hype about personalized medicine that is very media-driven. I mean, all medicine is personalized. If you go into the doctor with a bacterial infection, the doctor is not going to put a cast on your leg. By definition, medicine is personalized. But there's a movement towards hyper-personalized medicine. And while this is scientifically extraordinarily interesting and leading to very valuable discoveries, the implementation of that approach in medical care is beyond my understanding. Even in countries that have adequate healthcare systems, instead of the situation we have here in the United States, where doctors are increasingly having to spend less and less time with their patients. So we're more interested in general principles. We also approach problems by looking at diseases as tools for asking questions. So it is a perfectly valid and important part of the scientific path to decide that you are interested in multiple sclerosis or spinal cord injury or a particular kind of cancer. But there is another way to think about all these diseases, which is that different diseases allow you to ask different kinds of questions. We're also completely focused on drug repurposing. I'm going to talk about 3 different successful strategies that we have employed in order to make that more effective. Now, the first question is, which general principles are you going to work on? And we readily accept throughout the scientific community that it is perfectly fine to focus on RNA or gene or protein type questions, whether one works in humans or mice or Drosophila, C. elegans, any organism. And the reason for that is that these represent principles that were established very early in the course of evolution. So the way evolution works is that if something is sufficiently potent to provide a selective advantage, it becomes the foundation for everything else that is going to come. So nobody argues against this. So what I've been wondering in particular is, what about earlier stuff? This is all very, very late in the path to a cell. So let's go back in time another several hundred million years and focus attention on those general principles. The general principles that allowed the first steps on the road to cellular life. So you might ask, how do you figure out what those are? Well, the answer is that it's easy. You just think about it. You think about, what does a cell need? There are certain things that are absolute necessities. There has to be electron transfer. That is bioenergetics. That is all chemical interactions. Electron transfer is basically redox biology, the transfer of electrons from molecules with more of a reducing potential to ones with more of an oxidizing potential. There's pH. pH does many things, but one of the things that it does is it influences the probability of electron transfer reactions. There's nutrient supply. Without it, you don't grow. There's having an inside and an outside, because that is how you can control diffusion. That is how you create differences between the inside and the outside, and it is those differences in ion concentrations that allow you to generate electromotive forces that allow you to do electron transfer. So there's other rules that we can come up with, but these are ones that we have begun to test. And the hypothesis is that by focusing attention on these kinds of principles, we can get very powerful new insights into not only normal development, but also pathology. I've been trying to convince a couple of my mathematical colleagues to model the problem that the hypothesis that pathology does not confer a selective advantage except in the short term, and it will always lose out. And to me, that's a kind of an intuitive idea. So the way that I think of this is that what we're trying to do is understand what goes wrong in pathology at these fundamental levels and then fix it. Now, I wouldn't say anything like this if I didn't think that I had some hope of convincing you that this is not an unreasonable approach. We also think, and if you think about this from an evolutionary perspective, that much of what goes on here, maybe all of what goes on here, is subservient to this stuff. Certainly in the earliest stages of evolution, anything that happened here would not even confer a selective advantage unless it conferred a selective advantage in the context of this kind of stuff. So let's see how that works out. First part of the talk, and in various places in the talk, I'll talk about a cell called the oligodendrocyte type 2 astrocyte progenitor cell. Some people call it the oligodendrocyte precursor cell. It was one of the first progenitor cells that we had in our hands as a cell that we could isolate and know precisely what it was. Martin Raff and Bob Miller and I co-discovered it in 1983, and it's been a great cell for working out a lot of general paradigms. The cell divides as a progenitor or differentiates into a non-dividing oligodendrocyte. We discovered early on that in studying the oligodendrocytes, that redox biology was very fundamental in controlling their survival. We were among the first groups to discover that you could use antioxidants to prevent various types of cell death. And what I find endlessly puzzling and bemusing is that this paper is very highly cited, and so far as I can tell, no one pays any attention to what we thought was interesting in the paper, which is that small changes in redox state can turn on or off entire signaling systems. So if you expose oligodendrocytes to tumor necrosis factor, they die. If you increase their reduced state by about 20%, you can't kill them with tumor necrosis factor. If you give cells neurons a level of nerve growth factor that rescues 10 neurons, and you make them about 15 to 20% more reduced, now you have 1,000 neurons in the culture. So how in the world does that redox change, that small redox change, get converted into such a large biological output? When we then turned to studying progenitor cells, we found that similarly small changes in redox state control the probability of undergoing self-renewal versus cell cycle exit. I want to stress that we're interested in redox as a part of normal cellular regulation. So we don't work very much on oxidative stress, which is where most people in the field focus. Now, I mentioned that we had discovered that small changes in redox state have large effects on signaling efficacy. We also discovered that multiple signaling ligands necessarily alter redox state. So, for example, thyroid hormone is probably the best studied differentiation agent in biology. Everybody knows how it works. It controls gene transcriptional activity. Except that when we expose O2A progenitor cells to thyroid hormone, we make them just a little bit more oxidized, and if we antagonize that oxidation, we eliminate the effects of thyroid hormone on differentiation. Neurotrophin-3 is a classic agonist for a classic receptor tyrosine kinase that certainly causes phosphorylation cascades and the like. But it makes cells more reduced. And if we prevent the effects of neurotrophin-3 at making cells more reduced, we eliminate the effects of neurotrophin-3 on regulating the balance between self-renewal and differentiation. So how does this work? I had a very talented postdoc in the lab by the name of Zybo Lee, who agreed to take on the challenge of dissecting platelet-derived growth factor signaling from the nucleus back to the membrane in cells that were challenged with mild pro-oxidative stimuli. In this case, with low levels of several environmental toxicants, methylmercury, lead, and paraquat, although these rules apply to all pro-oxidants we've studied. What Zybo found was that when he oxidized progenitor cells, I should say the reason we work on PDGF is because it is the key basal mitogen for O2A progenitor cells. So when Zybo challenged cells with a pro-oxidant, he found that serum response element and NF-kappa B transcription were turned down, that ERKs and AKT phosphorylation was turned down, receptor phosphorylation was decreased, and that was why all of my friends in the signaling field and the stem cell field had been telling me for years that I should just drop this line of research, because, including not a few grant reviewers, that the view was that any protein with a cysteine on it is a redox target. And that makes this an insoluble problem, that redox affects everything. Well, we were fortunate enough to choose the correct control, and we chose neurotrophin-3, and what we discovered was that oxidation had no effect whatsoever on neurotrophin-3 signaling. So the idea that this was a non-specific regulator of everything was wrong. It was just wrong. So how do we get specificity? Well, Zybo saw in his gels that not only was the level of phosphorylated receptor decreased, but the level of total receptor was decreased. That was not caused by changes in transcription. So we went to the literature and asked, how is SIBL regulated? Sorry, how is PDGF receptor regulated? And it is one of the proteins that is regulated by an E3 ubiquitin ligase by the name of SIBL. SIBL is a member of a family of genes that, when they were first discovered, were given the wonderfully audacious name of ring proteins, where ring stands for really interesting new gene. And it's just great for someone to be so self-confident as to do something like that. But there's a lot of ring proteins. Turns out, however, that the general feeling in the field is that SIBL is the Lord of the Rings. And you're going to see part of the reason why. Okay, canonical SIBL activation is a way of controlling, of turning off receptor tyrosine kinase signaling. So ligand binds to the receptor is phosphorylated. That puts ubiquitin, that activates SIBL, which puts ubiquitin on the protein, and it's degraded. What we discovered is a non-canonical pathway. What Zybo learned from the immunology literature was that SIBL can also be activated by Fyn kinase. And there were 3 papers in the literature that said that Fyn kinase could be activated by oxidation. Put it together, and you have a hypothetical connection of all our results. And indeed, that's what happens. You oxidize a cell, you activate Fyn, you activate SIBL, put on ubiquitin, protein's degraded, all downstream signaling is suppressed. So, while most people study the interactions between Sybil and an individual receptor in which they are interested. We don't study that. We study pan-activation of Sybil. And that gives us the ability to talk to a lot of interesting proteins through this redox-Fin-Sybil pathway and to control their degradation and to control a lot of downstream functions. This will be my last slide on basic biology for this talk. But I just want to close the circle on thyroid hormone. Thyroid hormone and work done by Chris Foltz and Ibro Ambeskovich. We know it oxidizes cells. What they discovered is that it does so by opening mitochondrial pores. If we overexpress BCL2 or treat cells with cyclosporin A, we eliminate all effects of thyroid hormone on differentiation. If we knock out FIN or if we knock out Sybil, we eliminate all effects of thyroid hormone on cell cycle exit and differentiation. So thyroid hormone works through this redox-Fin-Sybil pathway. And what I'd like to propose to you just as a hypothesis to consider is that one of the big problems in cell biology is that we think we're so clever when we expose cells to two signaling molecules at once. But in the real world, cells are exposed to many, many things at the same time, giving conflicting information. Should I divide? Should I die? Should I differentiate? And the cell figures out what to do. Many of those signals we know converge on redox state. And if redox state all converges on the redox-Fin-Sybil pathway, you can control a lot of these decisions just with that simple means of summarizing the information the cell is getting. A hypothesis to be pursued. Okay. Now I'm going to switch to clinical questions. And first, take the redox-Fin-Sybil pathway into our work on developing new treatments for cancer. So we're interested in the same things as anyone else in cancer biology. We want to treat established tumors. We want to get rid of the cancer stem cells. We want to increase sensitivity to therapeutic agents. But we have another problem that we're interested in, which is that of the neurological damage done by many of our treatments for cancer. I noted about a decade ago that the clinical literature on neurological side effects of systemic chemotherapy had reached a point where you could not ignore it. This is a very serious problem affecting a lot of people. And nobody was studying this. So I had a few people in the lab who were interested in this, Jörg Dietrich and Rulan Han and Miranda Yang. We decided to go after this. We published 2 papers, about 2006 and 2008, and with those 2 papers, we doubled the worldwide literature. And this got covered by newspapers around the world. And what we established was that many anti-cancer agents are more toxic for progenitor cells of the brain and oligodendrocytes than they are for cancer cells. So our fundamental approach to thinking about cancer is always based on the question of how can we develop treatments that are not going to have such toxic side effects. And the hypothesis was that parallel analysis of cancer cells and primary cells would let us discover cancer-specific vulnerabilities. Well, a good hypothesis is part of the story, but it means nothing unless you are fortunate enough to be able to work with really wonderful, talented people. And I was extraordinarily fortunate that Brett Stevens joined the lab at this time. And Brett made some wonderful discoveries. So when we look at the proteins that drive things like tumor initiation and tumor growth, there's a big list. This is just a partial list of proteins that are important in these processes. And what's striking about this list is that everything on this line is a Sybil target. So question is, can we go after these? And things on this line are indirect targets of the things that are targeted by Sybil. Well, the problem is that if you want to activate Sybil in cancer cells, through the canonical pathway, it's really not of very much use because you're going to degrade that single receptor to which Sybil binds, and then the story's over. So the value of doing that in a cancer cell, theoretically, would be potentially interesting, but probably not of therapeutic value. But we have the oxidation pathway that allows us to pan-activate Sybil. So the question was, what oxidation agent should we use? And what we chose was tamoxifen. The reason we did that was because tamoxifen was the least toxic anti-cancer agent we had seen. It causes some damage in the brain. And tamoxifen kills cells by estrogen receptor-independent mechanisms. So yes, the anti-estrogenic effects of tamoxifen in treating early-stage luminal breast cancer are certainly of interest, but at higher concentrations, which are achieved within the tumor, now tamoxifen acts independently of the estrogen receptor, kills cells, and one of the ways in which it does that is by oxidizing them. So we had a good pro-oxidant that is probably the most widely used anti-cancer agent in the world. So Brett said, okay, let's activate the redox-Fin-Sybil pathway in glioblastoma cells, except the problem was that he could oxidize cells and he could activate Fin, but he couldn't activate Sybil. And what he discovered was that Sybil in these cells is complex with a protein called COOL1, or beta-PIX. It's a protein that not a lot of people study, and that virtually nobody in cancer studies. So Brett knocked down COOL1 and found that a lot of good things happened. That we could now downregulate the EGF receptor, Notch1 and beta-catenin are also Sybil targets. And in these experiments, Brett included as a control SOX2, a very important regulatory protein for many cancer cells, but it's not a Sybil target, except that when Brett knocked down COOL1, levels of SOX2 also decreased. When we look at tumor-initiating cells in vitro in a neurosphere assay, you get a few spheres, but they look like this instead of like this. But most importantly, you can't make a tumor. And if you inject these highly tumorigenic human glioblastoma cells in NSG mice, these cells will readily form tumors that look just like human glioblastoma, but not if COOL1 is knocked down. The only time you get tumors is if you inject a lot of cells, and then you'll get a rare tumor, and every single one of those tumors that Brett studied re-expressed COOL1. And he also showed that if you take the COOL1 knockdown cells and knock down Sybil in them, you restore tumor forming capacity, so the effects are Sybil dependent. Okay, but that doesn't get us into the clinic. We need drugs. And this is where we use one of our 3 drug discovery strategies. In this case, we're going to talk about a condition where we know a pathway and we want to do drug discovery. So unfortunately, you know enough about this pathway to be horrified by the prospects of drug discovery. There are no drugs that people have reported on for activating Sybil. COOL1 isn't studied at all, so there was no literature there on how to go after a COOL1 regulatory drug. And going after drugs that target protein-protein interactions is one of the most difficult problems in drug discovery. So we approached it in a very different way, which was to develop a screen that would tell us if Sybil was activated in glioblastoma cells. And we developed a strategy that's the equivalent of radiotriangulation, where we come in from 3 independent angles, 3 independent vectors that we think can only meet in one place. In this case, that can only meet on the activation of Sybil. And in this vector, Brett, out of 1,040 drugs screened from a library consisting mostly of already approved drugs, Brett found about 100 drugs, found about 100 drugs on this screen. This screen was to make sure we were looking at transcriptional inhibitors. There are about 900 drugs on that screen. Put them all together, and we're left with 10 drugs. So that's a very workable list. And the ones that Brett started looking at look to restore the ability to activate Sybil. And this is where we are now in terms of this work that has been carried on for the past few years by Jennifer Strapay in the lab, who's now moved to St. Jude. Blue is what we've validated. So we've hit a number of direct Sybil targets. We've expanded the list of indirect targets in a way that speaks to a much more interesting Sybil regulatory circuitry than had been previously recognized. So not only do we have NF-kappa B and serum response element and SOX2, we also have FOXM1, HSF1, HSP70, and I think everything else on the board is a given. That all of these are direct Sybil targets. All of these are targets of things that are up here. And in fact, we have many, many more targets besides these that Jen has discovered. And we do a lot of good, but the main thing comes when you actually test the therapies in vivo. So here we're taking 500,000 glioblastoma cells, putting them in intracranially in NSG mice, and letting them grow. They express luciferase, we let them grow for 2 or 3 weeks until we can see a tumor. So we're only treating cells where we know that animals where we know there's a tumor. And we treat with saline, or we treat with temozolomide, our frontline therapy for treating glioblastoma today, or with our drug treatment. So if we treat with saline, the animals live about 9 weeks, and that's when we stop all treatments. If we treat with temozolomide, the animals survive a few weeks longer, but the tumor is already growing during the time of treatment. For our therapy, we combine our frontline drug, which is an antidepressant, so we know it gets into the brain. It doesn't matter that it's an antidepressant. No other antidepressants came up on this list. It's just a chemical structure, but it gets into the brain. So the antidepressant plus tamoxifen plus temozolomide, because when you go into a clinical trial, you want to include standard of care. And when we do the triple treatment, there's no tumor growth during the time of treatment, and instead of extending lifespan by 20, 30%, we extend it by about 200%. If you look at tumor growth, this is the rate of growth in saline-treated animals on a log scale. In the temozolomide-treated animals, it's flat for a while, but in silver line, and then it starts to grow, and it grows just as fast. In the triple combo treated animals, there's no growth. Out here, this is a 3-fold increase from starting point as compared with a couple of 1,000-fold increase. With this glioblastoma cell line, there is no recurrence. Warner Chen was actually, as I recall, recruited to the lab by Brett. Is that right? And Warner took this on in basal-like breast cancers, another tumor group for whom we have very poor therapeutic options. And what Warner discovered is that these cells also inhibit Sybil, but they do it through a different pathway that is regulated by CDC42. Unfortunately, CDC42 does not complex with Sybil, and we don't know what the partner is yet. Warner's work, in particular, made this important contribution to our thinking. This therapeutic approach that we've developed is a two-key approach by necessity. That if you simply restore the ability to activate Sybil, that's not good enough. You might get rid of tumor stem cells, and that's what all of our data indicates, but in terms of harnessing it therapeutically, it's not good enough because the cancer cells have many mechanisms of controlling their oxidative state. If you just oxidize the cancer cells, they don't care. We can take cancer cells and make them so that 50% of their glutathione is oxidized. In normal cells, if you go above about 2% oxidized glutathione, you die. Cancer cells don't care. You have to do both. And indeed, in these experiments on basal-like breast cancer cells, this was a standard basal line implanted in mammary fat pads. This is how it grows in saline-treated animals. Grows about the same in tamoxifen-treated animals. This is an experimental CDC42 inhibitor. Grows about the same, but put them together, and now we're actually suppressing tumor growth of basal-like breast cancer cells with tamoxifen. And we hope that some of the drugs that were discovered by Warner are going to be much more effective. We're looking at that now. A very nice outcome of this approach is that in standard anti-cancer therapies, the progenitor cells are the most vulnerable cells, followed by the bulk tumor cells, followed by the cancer stem cells, which are resistant to many therapies. In contrast, this approach targets the cancer stem cells most effectively, followed by the bulk tumor cells, followed by the normal cells, and the safety profiles in the brain thus far look really good. We are far away from the world of mutation-based targeting, mutation-directed targeting. None of our proteins are mutated. We think we can predict responsiveness to treatment based on whether or not cells express a COOL1 Sybil complex. There are indications in that from Brett's work, and we're following that up now, because that's what you want to do in a clinical trial, is you want to stratify your patients to predict those most likely to respond. Our drugs are not only repurposed, they're all generic. We have good indications for multiple cancers. There's this selectivity. And the trials are very inexpensive because all we're doing is adding a drug to standard of care. So the insurance company pays for virtually everything. And the drugs we're talking about are so cheap that when our neuro-oncologist asked who was going to pay for them, I just said, I don't care, I'll pay for them. I mean, it doesn't matter. It's not a big expense. Okay, so now we've done pretty good on our radix work. We've gone from really fundamental principles in radix biology all the way to a very difficult clinical problem with a therapy that is poised for entry into clinical trials. So were we lucky? And certainly any kind of scientific success is based on the luck of getting people like Brett to work with you, to having colleagues like Craig who you can talk with about your science. But we wondered about whether we actually had a strategy. So we said, let's take on another big problem. Let's take on lysosomal dysfunction. We thought we were working on lysosomal dysfunction because of nutrient supply, to go back to our original list. That turned out to be, well, probably true, but not the key element. And here we're doing something very different. Here, we don't know anything about pathways. So what we do is work on the biology, use that to discover drugs, and then discover the pathways. Lysosomal dysfunction is important in a lot of diseases. The good Dr. Potter and I were having a great discussion about Alzheimer's disease. Parkinson's disease is important. Diabetes, macular degeneration. The list is growing, and I'm sorry to say it affects all of you because lysosomal dysfunction appears to be an inescapable consequence of aging. So, how are we going to study this? Well, we thought, let's go after the worst of these diseases, the ones that we know are caused by mutations in lysosomal enzymes. These are the lysosomal storage disorders. They're rare, but they're bad. And a lot of these diseases kill kids within 2 or 3 years. So our hope was that if we could discover strategies that were effective in these highly severe disorders, those strategies could be applied to other important diseases, but that were less severe in their phenotypes. So we focused in these diseases on a group called the sphingolipidoses, which are disorders of single lipid metabolism. And they are characterized by accumulation of substances that are thought to be somehow, in some way, toxic, although unknown why. And we started out particularly with a disease called Krabbe disease, unless you're German, in which case it's Krabbe disease, or if you come from somewhere else, it's Crab disease. It's a disease caused by mutation in galactocerebrosidase. There is accumulation of a particular lipid called psychosine, which had been shown to be toxic. But the state of the field for this and other disorders was very unsatisfying. It was that genetic mutations cause accumulation of stuff, multiple substances, and somehow that leads to multiple lysosomal and cellular dysfunctions. Not a hypothesis that gives rise to a lot of experimental design features. So we do what we do. We asked, what's the most vulnerable cell? Cellular and molecular foundations of toxicity, drug discovery. And this is work that was done by Christopher Fultz and Nicole Scott Hewitt. When we started taking this apart, we realized what an enormous challenge we had taken on. Because along with the multiple toxicities that had been reported, we discovered still others. And we found that the O2A progenitor cell was the most vulnerable cell. That was interesting because in this particular disease, there's a lot of myelin destruction and no repair. And in these progenitor cells, at sublethal levels of exposure, signaling pathways are torn apart, calcium's released, there's tremendous oxidative stress, the cytoskeleton collapses. So we thought that when we would apply drug discovery here, we might get drugs that beneficially affect one or two of these outcomes, and then we can make a cocktail. But we hit the jackpot. Chris hit the jackpot. He discovered drugs that blocked all toxicity. And that immediately suggested that he had discovered that there was a very, very important target that we could go after. So we did two things in parallel. First, we had a drug that was CNS penetrant, so we studied it in vivo, and we went after mechanisms. In vivo, and then we published this in PLOS Biology in December. So the drug that we published on is called NKH477. It's a forskolin derivative. And the reason why that's important, I'll get to in a minute. But we also found other drugs like an antibiotic, clofactol, and other things that were chemically different, functionally different. But anyway, we give this in vivo in these mice. This is a day 35 mouse. In the untreated mouse, the number of dividing progenitor cells is down by half. We rescue them. The amount of myelin is down by half when we rescue that. Motor function is severely compromised in these mice. This is just a beam walk analysis. We rescue that. And we extend survival as much as you do with gene therapy. Except the gene therapy has to be applied within 1 or 2 days after birth. And gene therapy works 24/7. We don't start our treatment until 10 days after birth because we know there's a lot of variability of onset in this disease, and we don't want to start until we can see symptoms. So we start at 10 days after birth, and we only give it once a day. And we're not correcting the gene defect, and our outcomes are already as good as gene therapy. So now we're working on ways to make them still better. Now, Nicole looked at the structures of lipids that accumulate in different sphingolipidoses and made a discovery that had evaded this field forever, that nobody else had recognized this, which is amazing because she did that as a second-year graduate student. So what she recognized was that among the lipids that accumulate in these diseases, each of them had a sphingosine derivative. And that sphingosine derivative had a free amino group. So what Nicole hypothesized was that when that lipid is trapped in the lysosome, the amino group would become protonated, and that would alkalize the lysosome. And a lysosomal function is dependent upon having an acidic pH. So that's a very testable hypothesis, particularly because one of the proteins that controls lysosomal reacidification is the cystic fibrosis transmembrane receptor. And the CFTR has been so well studied that there's good agonist drugs and there's good inhibitor drugs to use. And indeed, we found that we prevent all toxicities by activating the cystic fibrosis transmembrane receptor. We found that all the drugs that Chris had discovered converge on requiring activation of protein kinase A, and protein kinase A activates the CFTR. So our view with the lysosomal storage disorders, at least the sphingolipidoses now, is a little different. It's that it's not just accumulation of stuff that somehow causes all these dysfunctions. It's that there is accumulation of very specific lysosphingolipids that increase the lysosomal pH, and that causes everything else. All the other toxicities are downstream of this. And if we come in with a CFTR agonist or with our protective agents, we prevent the lysosomal pH increases, and we rescue everything. So it's a very different view of not just these diseases, but in the hierarchy of lysosomal dysfunctions. And that area, too, is moving on in interesting ways in the group. So we entered this thinking we were going to study nutrient supply. In fact, we ended up studying pH. So back to one of these early regulators. Very, very short, just a couple minutes on inside, outside, and controlling diffusion. So now we're going to deal with a very different problem. And this is drug repurposing by first principles, by looking at a problem, knowing what a drug does, and now applying it in a novel way. I became interested in this work, which I confess I had largely paid no attention to over the years, because I was fortunate enough that there was an orthopedic surgeon at Rochester by the name of John L Farr who had discovered that if he crushed the sciatic nerve and injected animals with erythropoietin, they got better faster. And the department chair asked if I would help mentor him because they're orthopedists and don't know any neuroscience. And I said, sure. So I met with L Farr, and he's a great guy, really smart and passionate, and a really good teacher. And he started teaching me about peripheral nerve injury. So the problem in peripheral nerve injury, the central problem, is that the surgeon comes and looks at their patient, and the patient has lost motor function or sensory function or both. The surgeon has to make a decision. If the nerve is damaged but still has some integrity, that there's still some axons that project through the lesion site, you leave it alone because there will be a significant amount of spontaneous recovery in many patients. If the nerve has been cut, you have to go in and stitch the ends together. But if you do that operation in someone whose nerve had some integrity, you are now going to eliminate that integrity and you're going to eliminate the spontaneous recovery, which is not a good outcome. So you need a diagnostic criteria for making this decision. And the diagnostic criteria as it exists right now is watch. Sometimes for weeks, sometimes for months. And if at the end of that time, the patient gets better spontaneously, you feel good, you made the right decision, you didn't intervene. And if the patient doesn't get better spontaneously, you say, oh my goodness, I should have done surgery, but now it's too late. And that's where this field has been for decades. And that's, I think, why all the many papers that you might read on ways of enhancing peripheral nerve regeneration don't make it into clinical trials, because you can't get beyond this first hurdle. So, what happens when a nerve is damaged but still present is that the myelin can get damaged. And when the myelin is damaged, potassium channels leak out. Potassium ions leak out. And there's a drug that has been studied since the late 1970s called 4-aminopyridine that blocks those potassium channels. And it has been recognized since the early 1980s that this drug enables demyelinated axons to conduct impulses. And people have studied this drug in many chronic neurological injuries. And it's actually approved for treating patients with multiple sclerosis. The situation in chronic injuries is that you treat with the drug, you get symptomatic relief. If you stop treating with the drug, all the benefits go away. So there's no durable benefits. Now, we went into this first asking what would happen if we applied it in the acute setting to see if we could do a diagnosis, which amazingly no one had ever done. And when we take a crushed lesion and treat with 4-aminopyridine, we get better function. When we do this with a cut injury, there's no improvement in function. So, and this drug works very fast in humans also. So this is something we may be able to do diagnosis a day or two after injury. When we gave it every day, we discovered that we caused durable increases in function. That the animals got better in motor function faster, their nerve conduction velocity increased faster and increased more than occurred in spontaneous recovery. Nerve conduction velocity is controlled by remyelination. And indeed, we discovered that 4-aminopyridine treatment promotes remyelination in these injuries. So this is just looking at one myelin protein called P0. It comes back faster in the treated animals. If we use one of the standard measures of myelination, which is the ratio of the myelin area to the axon area. It's called G ratio. When you have an injury, it moves away from the normal distribution. In the treated animals, it's back to the normal distribution. So what we see here is that with crush injury, you get decreased motor function, decreased nerve conduction, demyelination. When we treat with 4-aminopyridine, we restore motor function and nerve conduction, and we promote remyelination. So that's inside-outside, right? What's going on is that you're breaking the normal means by which you keep the inside different from the outside. You're perturbing the ionic balance. All we're doing is restoring it to normal. So this general hypothesis actually seems to be not bad. And instead of 5% of our discoveries looking like they can move to the clinic, all of our discoveries look like they're going to move to the clinic. The FDA immediately approved our first clinical trial proposal for 4-aminopyridine in peripheral nerve injury. We have to work on the IND submission for the glioblastoma trial, but I think that'll go very well. And the lysosomal work, we're just trying to figure out what a clinical trial might best look like. So 3 different strategies for drug discovery based on this fundamental hypothesis, clinical trials moving forward, application to other cancers looks pretty good. And now the people. I've had the honor of working with Margot Meyer-Purcell and Chris Purcell since 1990. We started working together in London. We moved together to Utah. We moved together in Rochester. We're all independent scientists with our own labs, but we function as basically as a socialist collective where the good of the group is to the benefit of everybody. And there's many different projects, many different approaches, many different ideas. Most important, however, are the people who do me the honor of coming to work in the lab. So I mentioned Zybo Lee, who discovered the Radox-Fin-Sybil pathway. Chris Foltz, Nicole Scott Hewitt, was critical in the lysosomal storage disorders. Jörg Dietrich, Rulon Hahn, Miranda Yang, and Anna Nunes on the effects of chemotherapy on the brain. Sybil function, the stars are definitely Brett Stevens, Chris Foltz, Warner Chen. Sorry, Brett, I didn't bold your name because I had used this slide at a Harvard talk where Warner was in the audience. Brett's name also should be bolded. And Jen Strapay, of course, has been doing the drug work, and the 4-aminopyridine work has been done with John L Farr and Chris Sang. There's been a lot of people who have generously supported this work. So that's us, and I'd be delighted to take any questions. Yeah. So the question is that the mice who were treated with our simple restorative agent, how is their cognitive function? So they can still read. I've noticed changes in their television channel preferences. And we don't, I don't know. But we haven't looked at. They look really healthy. And when we look at their brains, we're not seeing a lot of cell death or even decreases in precursor proliferation. So we're hopeful it'll be good, but we'll see. Yes? Right. Absolutely. Great. And for those of you who do not understand Andrew's deep Parisian accent, that the question is, basically, how does this work in a way? I mean, we're talking about proteins that are important in the function of every cell, and particularly precursor cells. So how in the world can you get any kind of selectivity out of this? And hypothetically, an answer that attracts us is that in normal cells, the redox-fin-Sybil pathway is on. It works. So these toxic agents are going to do what they do. If you restore Sybil function, it doesn't matter because Sybil's already functional, so you're not changing anything. But in the cancer cells, you're dependent on that Sybil inhibition. So the cancer cells, at least the ones we've been studying, absolutely require that. And is it the case that we're getting therapeutic benefit because we're getting 40 to 70% inhibition of a whole multitude of proteins? And that if we did that experiment in O2A progenitor cells, we would find that they're actually quite happy? Well, we're gearing up to look at that in the lab. That's the outcome that we would like, that Andrew and I were having an interesting discussion yesterday on the idea that, which also Craig and I have talked about many times, that despite their ability to be psychokillers in vivo, cancer cells are actually very fragile little beasties, and actually much more fragile than normal cells. So we'll see. This is something that we're working on, but absolutely it's the right question. Yeah. Yeah. Well, we know that it's in a complex with Sybil from the pull downs, and we're finally going after this in a proteomics-based experimentation now. So I hope in a couple of weeks I'd be able to give you a good answer to that question. All we know is the complex exists, and we don't do any experiments on overexpressing proteins and looking at the biochemistry because there's a lot of people who are way better at that stuff than we are. That's right. Well, I actually think they're similar because within the central nervous system, there's a couple papers that show that axonal conduction, axonal impulse activity, promotes early myelination during development. And there's a couple of less well-developed suggestions of that for peripheral myelination of Schwann cells. So the working hypothesis is that what we are doing is that we are basically recapitulating this developmental strategy. It's a hard question to ask functionally because if you disrupt neuronal impulse conduction, of course, you're not going to see anything. So, and one of the advantages of working on clinically directed questions where your real goal is to get out of the lab and into the clinic is that you have the luxury of foregoing a lot of these very important but complicated mechanistic analysis. An opportunity I highly recommend to people.
So, Mark, earlier in your talk, you alluded to this fascinating observation that redox state itself has a real potential in stem cell populations closely linked. And I believe you were the first to show that activity is subject to this has been shown in, I believe, every somatic stem cell type that anybody's ever looked at. It's also true in at least breast cancer. Mm-hmm. So the property depends on unbelievably well observed. The question is, have you or anybody else tried to link some kind of molecular underpinning to that in the same way you have Sybil and we think why that?
I mean, the way that I think about that question is that things like redox signaling is what you had at the beginning. So the reason why it's linked is because everything was built on top of that. So then the question is, how is it linked? Well, we thought we were going to discover this incredibly complicated system, but FIN itself appears to be directly activated by oxidized glutathione. So it could be that this is a case where the circuitry is extremely simple. Now, how does reduction work? Well, reduction, we know in part suppresses Sybil activation. And therefore you keep high the levels of these various receptors and of ERK phosphorylation and AKT phosphorylation, NF-kappa B activities, maybe SOX2, FOXM1, things like this. So we think that's how you would promote self-renewal. One of the questions that comes out of this is that there are some stem cells, like the hematopoietic stem cells, and from work from Harley Kornblum's lab, the neuroepithelial stem cells of the subventricular zone, that are triggered to divide when they're oxidized. And then they transition to a state where they're inhibited to divide when they're oxidized. And, we wonder whether an oxidative burst is a part of the primitive response to damage signals and gives you a universal ability to call stem cells into action. If that's the case, or just with the case that some stem cells will divide when oxidized, of course what we want to know is whether they're using the same strategies the cancer cells are, and we'll find that out. We have some hints that might be the case.
Another question. Thank you again, Mark.
Thank you.